lighting is okay if it's too dark. And in fact, since it looks so far as if it's cracked the issue this time, and I apologise almost to excess for last time. It was a really unprofessional amateur show, so sorry about that. But it looks like we cracked it this time, so we can go on. And this time, because we're starting in a timely manner, on time. If you have any questions as I go along, please don't hesitate to ask them. It simply means that I haven't explained it clearly enough. And if you ask a question, I mean, then hopefully we can all learn from the answer, if I indeed know the answer. One final thing is to, before I forget, is to give a plug for tomorrow's departmental DLS seminar, which is going to be given by Brian Lamb from the Department of Clinical Biochemistry at the University of Cambridge, and he's going to be talking about spatial mapping of hypothalamic neurons, so taking a transcriptomic approach. So those of you who are interested in neurobiology or bioinformatics and related subjects, and also he's working on metabolism, so he's interested in so he covers lots of bases. He's a very, very smart bloke, and I do recommend that you come along. And I've asked, I don't know if this is going to happen, but I've asked if we can extend a little bit beyond, particularly for you guys and people who might like to talk to him, so we can have a Q&A where you can ask him questions about career-y things and how to get into this if you're interested in it and stuff like that. So I don't know if that will happen, but that's certainly my intention, and I very much hope it does. But today, what we're going to be talking about is, here's an overview of the lecture. On Thursday we were talking about the cell cycle mainly, how the cell cycle changes because there's a signal from the sperm that induces meiotic exit. And while that's going on in many cases, there's all of this other stuff going on that we're going to be talking about today, which is mainly on the kind of chromatin remodeling, so how the genomes are programmed or reprogrammed to participate in embryonic development. So that's what I want to talk about today, leading up to embryonic genome activation, because one of the points here is that these transcriptionally inert gametes, the sperm and the egg, they're transcriptionally silent. There's no transcription going on, almost no transcription going on, and that has to be switched on somehow in the nascent embryo, the ones that we think happens at the one cell stage. And so how does that happen? So I want to talk a little bit about that, and I'll just finish with nuclear transfer cloning, because overall we're talking about the establishment of totipotency, the ability of a cell to give rise to an entire organism, an entire individual. And you do that, of course, canonically with fertilization, but also artificially, if I can use that term. You do it by nuclear transfer cloning, you can do it by nuclear transfer. So I'll come on to that at the end. So this is from last time, this is the kind of cell cycle changing, and during this cell cycle exchange, the reduction of meiosis, metaphase 2x, M2 means metaphase 2, sperm nuclear protein is exchanged. I want to say a little bit about that. So this is encapsulated in these terms here: decondensation, first of all, of the sperm chromatin or sperm nucleus, followed by recondensation. So decondensation, the first one of these, is accompanied by an increase in the chromatin volume. So we can visualize that, and the way we do it, although some of you might think it's a rather jaded and old-fashioned method, it's actually very useful, and it's immunofluorescence microscopy. And I just want to mention something about that in the context of studying sperm decondensation. So we're using confocal microscopy, immunofluorescence, so it's kind of indirect. We're using an antibody against epitopes of interest, which we can then label to visualize where those epitopes of interest are. And it's very useful, immunofluorescence, to visualize kind of global distribution of antigens or the epitope that you're interested in. So you characterize the antibody you're looking at, so you know from other experiments and people who sell antibodies that do this really in great detail, to show that your antibody is specific for the epitope that it says on the tin, right? Not going to cross-react with lots of things. That's in an ideal world. In practice, they often do cross-react, but ideally, you want an antibody that's specific. And so this is the kind of thing that you can see if we're looking at decondensation in sperm. So what this left-hand panel shows, these are paired images, so it's the same egg. And actually, in this egg, after 20 minutes of injecting a sperm into the egg, and there's no bright field here, so you have to kind of imagine that the egg is all this round here. And what the bottom row shows is staining for mainly genomic DNA with propidium iodide. There are different stains for DNA. This one is propidium iodide, so it shows in the red channel here. And these are this is 20 minutes after the sperm has been injected, and it shows so the maternal chromosomes, remember metaphase 2, so they're metaphase, they're condensed chromosomes of metaphase 2, and after 20 minutes, and the sperm. And in fact, we've done something here to this cell, which I won't go into because it's not important for now. And here's the sperm head. It's been injected actually, and you can see it's the crescent shape of the sperm head. And basically, it's all taken up with this nucleon, largely taken up with this nuclear DNA, so it's stained with propidium iodide. And now this is after 20 minutes at high magnification. And these panels to the right here, they're all the same. They're all paired images, vertically paired images at the sort of higher magnification, just to show what's going on with the sperm, because we're interested in decondensation. Okay. So this is after 20 minutes, and in the top row, we're looking at staining on the epitope, and the epitope is is an H1 epitope. H1 is a histone, it's a linker histone, and it's a variant actually. I won't talk about it, but it's a variant that's present in eggs, but it plays the same role from H1M00 for oocyte, and it basically is it's a linker histone, so it's something that's going to link the nucleosomes of nucleosomal chromatin. So you've got in your kind of classical model, you've got your 147 base pairs of DNA wrapped around the histone optima, so 2H2As, 2H2Bs, 2H3s and 2H4s, and they're linked together with an H1 linker. So what we're looking at is kind of the assembly of the paternal genome with the maternally derived histones marked by H1. So we're looking at the chromatin expanding, and I'll say what's happening as the expansion, because basically protein is being removed. But here, what we're looking at is the deposition. You can't see anything, there's no staining here, right, for H1. There's a little bit, if you use your imagination, so a little bit, very, very faintly there, and I'll come back to this, but it starts to acquire, and as it does this, it's all same scale. This is, I don't know what it is, probably 5 microns. It starts to expand after 80 minutes. It's expanded like that as the maternal histones go on, indicated here by that H1 stain. So, in order for this population of the paternal genome to happen, the resident nuclear proteins of the sperm, when the sperm goes in during fertilization, have to be removed. So, the major sperm nuclear protein is protamine. Okay, and that removal is one of the features of decondensation and expansion of the sperm volume. So here are a few bits of information about protamine. Much of it's incomplete, but this is some of the stuff that we know. So in sperm, and I'm talking about mouse mainly, but also humans. So in the mouse, about 98 to 99% of nuclear protein in sperm is thought to be protamine. Okay. In humans, it's probably closer to 85%, all right, and the remainder in each case is thought to be histone. Zebrafish lack protamine, some other animals lack protamine. I mentioned zebrafish because it's a model system, but not really a model system for this. And there are many, so I have these conversations with people who work on zebrafish, and it's okay. So cynically, I call it a model for zebrafish, really. So zebrafish is a great model for zebrafish. But anyway, so it's restricted, protamine is restricted in spermatids and sperm, so you don't find protamine outside of these cell types. So it's involved in spermatogenesis in sperm itself. They are arginine and cysteine rich basic proteins, and they are thought to form a toroidal, like donut structure with DNA wrapped around them, rather than the nucleosomal structure that I just mentioned. And that's thought to be very important because it allows a phenomenally dense packing. So there's very little solvent in the nucleus of the sperm. So it's got the highest packing density of any cell type. It's really extraordinary, and that's facilitated by these disulfide bonds. There's also cysteine-rich disulfide bonds. Now, protamines are, in mouse and human, encoded by 2 different genes, PRM1 and PRM2. So PRM1 is synthesized in its mature form and is 50 amino acids in length, and PRM2 is synthesized as a precursor that's processed to its mature 63 amino acid mature form, and it binds to a single zinc ion. So, yes, most of the chromatin in sperm, the nuclear protein in sperm, is protamine, but some of it is histone. And this is thought to be in a nucleosomal form, so the canonical octamers, and it's located at promoters. So this is something that people have realised started to characterise fairly recently with the advent of some of these sort of chromatin techniques. And it's thought to be located at developmentally important genes, and many of them contain trimethylated H3K4, so lysine 4, which is trimethylated, or K27 trimethylated, so lysine 27 trimethylated. Sorry, did I say H4? H3 in both cases. Histone 3, the fourth lysine is trimethylated. And in this case, for this mark, this is a mark that in promoter regions is generally associated with active chromatin, active transcription. Whereas this mark is generally associated in promoters with silent chromatin. So you've got a kind of yin-yang here, the push-pull thing here, that was first identified in ES cells and is thought to potentiate ES cells so it stops them from being, the genes from being transcribed. It's called a bivalent mark, but it poises them for transcription when differentiation starts. Remember, ES cells are derived from the inner cell mats, so they're going to start to differentiate into all of these different lineages. So it potentiates that. It's thought to potentiate that differentiation in ES cells and possibly also in sperm. So most, and this is a global thing, and it's a little bit contradictory. There's different information from different sources, but most of these genes are not expressed in pre-implantation embryos. In general, this mark is associated with PCG, so polycomb group pressor complex, mediated gene silencing. So that's facultative. Facultative means non-obligate. So it kind of sometimes it is, sometimes it isn't. Heterochromatin means it's kind of locked up and not generally transcribed. And euchromatin is the opposite. And the paternal, the sperm side, once it's acquired, this mark is generally globally depleted during pre-implantation development. That's just FYI. I'm not going to go into any more detail on that. I want to talk now a little bit though about the exchange. How does the exchange of proteins and the replacement largely with histones occur at the sperm? So this is this model which I showed you in small in the previous slide of showing the toroidal DNA wrapped up with the protamine. So this is what in sperm. So when the sperm goes in, this is what it looks like, and it has to be removed and replaced with maternal histones. And that's what decondensation really is. Protamine being removed and rapidly replaced with maternally derived histones. So if you think about the oocyte, the egg that's fertilized, it really is an extraordinary cell because it's got all of these components that are necessary to drive, necessary but not sufficient, to drive early embryogenesis. And the histones that are going to populate paternal DNA, chromosomes, are part of that pool that the embryo inherits from the egg. So all of this remodeling, occurs independently of DNA replication. That's something that makes it rather unique, actually. I mean, I shouldn't say that. It makes it unusual because normally this kind of remodeling happens after the replication fork in somatic cells, say, has passed. So it's during S phase. This is there's no S phase going on here. This is happening within minutes, as you saw, 80 minutes of the sperm entering the egg. So it's prior to the first S phase during interkinesis. And as I said, it's kind of unique. Perhaps I shouldn't say unique, but very unusual. Now, how does this happen in a slightly more detail? What are the molecules involved? And this has been worked out mostly, or initially, in Xenopus. The thing about Xenopus laevis in particular is, again, it's a model system that's it's not so popular now because the good thing about it is that you can do bucket biochemistry. You know, you can collect these eggs, for example, in literally buckets. It's not for everybody. But you can do that, so then you can purify stuff and you can work out what's going on. And these first chaperones or proteins that are removing protamine from sperm in fertilization were isolated, identified in the early 1990s in Cambridge, actually, in Xenopus. The trouble with Xenopus laevis is that it's a tetraploid, apart from that, there are many problems with it actually, but one big one is that it's a tetraploid organism. So doing genetics on Xenopus laevis is extremely non-trivial. And so people have turned to other systems rather than this. But back in the day, people were doing biochemistry on Xenopus, and it was a good system. So what they found in Xenopus is that the protamine is removed by a protein that's known as called nucleoplasmin, which is an acidic chaperone. It's widely expressed, so this is now talking about mammals as well, and I'll talk about them, because there are 3 structural counterparts that are conserved in mammals. So let's go through these very, very briefly, because it's not really all that edifying what I'm going to tell you, because we actually really still, that's a very fundamental process, we don't really still know how they work. And what I'm going to show you now is largely from 1 or 2 papers, the most recent of which was in nucleic acid research over 10 years ago. There's not that much has been added to this since. But it looks like for the 3 counterparts, the 3 species orthologs of nucleoplasmin, the first one, NPM1, nucleoplasmin 1, is actually called nucleophosmin. It's quite a large protein, and it, with NPM2 of the 3, mediates nucleosome assembly in the newly forming embryo, but also in vitro, and it has an RNA binding domain. So somehow RNA is involved in this, and there is a little bit of progress on this, which you can find out if you're very interested, using link RNA actually. So link RNA that seems to be involved in this, but I'm not going to talk about that now. It's also, like a lot of these proteins actually, is involved in cancer when they're translocated, So if there's a translocation that activates this gene ectopically in somatic cells, it can give rise to cancer, which isn't terribly surprising if you think that this protein is going to go in and remodel things. It's an acidic protein, so it's not necessarily just going to be targeted, to proteins. It might also target other basic proteins, like in some circumstances, histone. So in somatic cells, if it's upregulated and it's sort of the levels are kind of non-physiological, then it can give rise to cancer. NPM2, now there are null knockout mice that have been reported for this, and they have abnormal decondensation, abnormal decondensation, but abnormal pronuclei. So NPM2 seems to have a different and actually not fully understood role, far from fully understood role in decondensation, because if you make knockout mice and you use those oocytes, then they undergo the sperm. When you fertilize those oocytes, undergo what look like normal fertilization. They're not normal because the pronuclei are abnormal, but they look like there's no gross abnormality. NPM3, the third one, is a shorter protein, and it's involved, and it seems to mediate in vitro decondensation and nucleosome assembly with NPM1. So these things that work in concert. So they seem to work in concert. These three enzymes. So there are mammalian counterparts to the Xenopus nucleoplasmins, whose function and mechanism we don't really understand. So this is work in progress. So that's something that would be a really cool thing to do. If that's what you did with your life, you worked out how sperm decondensation works, how remodeling of sperm works in fertilization. That wouldn't be bad. Right? It's something we just don't know about. I think you might need some help, but it's a big job. So after this decondensation and the proteins come off, then there is nucleoprotein deposition. Okay, so decondensation is followed by recondensation, and this really is another way of saying, as far as the sperm is concerned, it's building the chromatin of the sperm. It's assembly. So this initiates starting about 20 minutes after sperm entry into the mouse, in mouse oocytes, as you saw, and it lasts several hours, and it might be primed by the sperm-borne histones. We don't really know. That they might actually give the signal for how to actually initiate this process. If it's true, it's a possible mechanism for transgenerational epigenetic inheritance, because you can imagine how in the sperm, at least, we're talking about from the paternal side, these chromatin, these histone nucleosomal islands have been influenced during gametogenesis, so during the parental, whatever the parent's doing, to give rise to epigenetic inheritance. And this might be one mechanism by which this happens, by these paternal, these histones, nucleosomes. And also it could be like protein, something you might think about. I don't know whether you've come across this idea of transgenerational epigenetic inheritance. Perhaps I should just say 1 or 2 words about it. So we all think about Darwinian evolution in terms of genetic, what's generally considered to be the genetic material in mammals, which is DNA. Some viruses have RNA. It's almost always DNA everywhere else. Double-stranded DNA. And so changes, mutations perhaps in the DNA sequence can give rise to different phenotypes that can be acted upon then by the environment, giving rise to the survival of the fittest. That's kind of Darwinian. That's the broadly speaking the idea of Darwinian inheritance and evolution. But it turns out, and about, I don't know, sort of 80 years before that, Jean-Baptiste Lamarck had come up with this idea, that giraffes get their long necks because they're reaching for the branches, and their necks get a little bit longer, and the next generation inherits that, and their necks get a little bit longer. And everyone laughed at that, and they still do to some extent. But it looks like, in part, there's some truth in this in some cases. So it looks like, increasingly looks like, for example, one trait which has a great deal of economic and clinical relevance is acquired obesity. So if the parents have a high BMI because they've been putting on weight because of what they've been consuming, then in the next generation, then their offspring also have obesogenic traits, even though their genetics are the same. So somehow that obesogenicity of mother or father is transmitted to the next generation. And you can show this in a mouse model using gametes. So you can feed up with high-fat diet the parents, and you can remove the gametes, do in vitro fertilization, put the gametes, the resulting embryo, back into a normally fed, normal chow dam, recipient mother, and the offspring will be obesogenic. So there's something going on here, and that's more like Lamarck, because this phenotype has been acquired. This is something like the long neck of the giraffe. And there are other examples of this to do with neurobiology as well. So it's something that's worth bearing in the back of your mind. It's interesting, and you definitely know how it works. So sperm nuclear proteins are exchanged, and these is for histones, for the maternal histones, and these include histone variants. So there are H2A isoforms, 2 of them here, and you can read this. I don't particularly want to dwell on it, because it doesn't really particularly lead anywhere here. And also there's an isoform of H3 called H3.3, and that's deposited by the chaperone HeRa, and that's true across in the metazoan, so that's true in flies, as well as it seems to be in mammals. So these variants are typical. By the way, when I say variants of H3, they vary by 1 or 2 amino acids. They're not big variations. Okay, so it's just small variations. And H3.3, in general terms, H3.3 deposition is DNA replication independent, so it's S-phase independent, which is appropriate here because there's no S-phase. Okay, so it's deposited in the paternal, mainly the paternal genome, somewhat in the maternal, and is present in these 2 different types, heterochromatin and euchromatin, but thereafter in pre-implantation development restricted to euchromatin. So there we have it. So both of the genomes, so the maternal genome had already been assembled, chromatin had already been assembled, it's present with metaphase chromosomes. The paternal chromatin has to be completely remodeled, get rid of the proteins, and repopulated DNA, the genome, with histones. So that's happened, or is happening, and now what happens is that they become encapsulated in membranes, and they're separate. So the different parental genomes are separate, the maternal one and the paternal one, in these things called pronuclei. So paternal, so what it says there, and this is all going in 1-cell embryos, and you can see this in the mouse after about 4 or 4.5 hours by sort of standard, bright field microscopy. And this is not a very good image actually of a mouse oocyte, mouse 1-cell embryo, about 6 hours after fertilization. And this is, it's labelled in a minute, but before the labels come on, I just want to show you this here. So this is a pronucleus, You can see this kind of clear region here, and the bit in the middle is a pronucleolus, so ribosome RNA genes. So they're both here, and the next one I'm going to show you the labels of this, so you can see the labels. And in the mouse, the male pronucleus is slightly larger than the female pronucleus. This is a degenerate polar body 1, first polar body, and this is a polar body 2 that's fragmented into 2, which is why this is a bad illustration, because it doesn't usually happen like that. So I've just said that, but the pronuclei are the same size as each other in maternal and paternal pronuclei are the same size as each other in humans. It's not really known exactly how pronuclei are formed or what from, but it's thought to be derived from the endoplasmic reticulum of the oocyte. Okay? It's not DNA sequence specific, so if you inject bacteriophage lambda DNA into an oocyte and then activate development artificially, or then it will cause pronucleus formation. So there's nothing particular. So, and that's small. So the genome of phage lambda is 48 kilobases, which compared to these mammalian genome sizes is tiny. So 48 kilobases will do the trick. You inject that DNA. So it's the assembly, of course, is required for nuclear pore complex formation, which occurs in pronuclei because they need to process RNA to get RNA out. And here you can see them; they're separate, and they gradually converge. They gradually come together, and that involves forces, which I'm not going to talk about here. We have actually, there's a paper at the end, Duce et al., with our collaborators in Spain. We actually injected nanodevices, which are huge in cellular terms. They're about 20 microns long and 10 microns wide, but very, very thin, only 20 nanometers thick, which is about the same thickness as a microtubule, so tubulin polymer. And these things, if you inject them, you can watch them. We injected actually, they're like H's, but with 4 prongs on each side. And if you inject them, you can watch these prongs deflect. And because they're made in a physics laboratory, we know exactly what forces corresponds to the extent of deflection. So we've been able to measure intracellularly, during development of the one-cell embryo, the forces that are acting on this thing. And they're amazing. There's a program of force changes, and these forces are involved in moving organelles like pronuclei around. So we've barely started to understand what these forces are, how they're generated, and how important they are. We can't really manipulate them. We're just starting to be able to study them and recognise that they're there. But anyway, they move together using these forces, and when they're together, there is nuclear envelope breakdown. So we go into M phase, okay, and that's mitotic prometaphase, M phase. So now we're in a mitotic cell, it becomes a mitotic cell. So we've exited meiosis, there's something called interkinesis, and now we go into this new cell cycle. It's a segue from meiotic cell cycle to the mitotic cell cycle, into mitotic really S phase, and now we're into mitotic M phase, where we're preparing for cytokinesis. And that's what happens just before that occurs. And when the membranes break down, the chromosomes can come together, and of course by then they've undergone S phase and cell division. So this is called, that coming together of the chromosomes is called singling. Now the histones in both parental chromatin sets have N-terminal modifications. So this is something that you'll probably be aware of. And some of these are controlled by the meiotic cell cycle progression. Actually, just let me take a step back very quickly. Are you aware of the epigenetic kind of the modifications that go on to the histone N-terminal tails? Because I've been talking about, say, H3, K4 trimethylation, as if you all have, and it's something that's a general principle in kind of the control of gene expression, for example, or DNA repair and other processes in cells, the somatic cells. But I just wonder, I mean, if you don't know about it, don't worry. But I think it's something you probably should look up and sort of get your heads around a little bit if this is really sounding like Greek to you, because it's general importance. So I do recommend that you understand a little bit about these epigenetic modifications which occur by modifying histones. And in eggs, and I'll say a little bit about these modifications. But in early embryos, they differ between the two parental chromosome sets because they can do that because they're packaged in these pronuclei. So I'm stepping a little bit back in time now from singling. So when they're packaged, you can have one set of modifications going on in one pronucleus and another set of modifications going on in the other one to give rise to different sort of gene expression, for example. And again, you can study this using immunofluorescence. And here's a whole bunch of it, same kind of principle as before, except that now we're using a different kind of dye to study the DNA, right? It's a general DNA dye, a different one. This is the first one, staining in blue. And again, they're vertically paired images. And now, instead of using antibodies against the linker histone H1, we're using antibodies against, and I thought it was unbelievable when I first heard about it, against specific N-terminal modifications of the histones indicated. So this one here, so these are all, so in each case, vertically paired case, it's the same embryo, so for this one here, it's stained with, for DNA, and here it's stained with an antibody against this mark that we've already talked about, trimethylation of H3 lysine 4, okay? And if you do that, it looks like, I'm guessing here, but there's more staining in the pronucleus that's maternal than there is in the paternal one. And these are being controlled for, so there's no H3, there's no phosphorylated serine 10 in H3. There looks like in both pronuclei, there is dimethylation of arginine 17 on H3, and so on and so on. But you can see that there are some differences. Some of them are marked. This is global staining, remember, in immunofluorescence. So that gives you a good global readout. And the amazing thing is that there are, to me, is that there are antibodies that are specific for these modifications. You could distinguish between H3K4 dimethylation and H3K4 trimethylation because there are antibodies that are specific to these very subtle differences, well, single nucleotide in this case. So these slides just summarise some of these changes, and I don't want to dwell on them because basically they are what they are, where they are. And I want to really just spend a little bit of time talking about newer methods for studying chromatin, even at the single cell level. So some of you will be aware of these, but I want to make you all at least aware of them now. Because immunofluorescence, which I think is fantastic, don't dismiss something because it's been around for a long time. Some of these things can be very, very powerful. And there's too much kind of sniffiness among some elitist scientists saying, oh, well, that's no good. They were doing that 10 years ago. But, well, yeah, sometimes that's true. I mean, you've got better methods, but sometimes the old methods still give you the information that you want. So that's the question: what information do you want? Then don't care about how you get it. So, but what I want to talk about now are some locus-specific high-resolution methods that do give you a different type of information. Because the things that we've been looking at are global. So supposing you want to know whether your favourite gene is bound by H3K4 trimethylated histone, right? So histone H3K4 is trimethylated. If you want to know whether your favourite gene is bound, then you can't do it using immunofluorescence. So now you can do chromatin analysis at high resolution, previously with something called chromatin immunoprecipitation, sequencing and what you pull down. So essentially you have your antibody against the thing that you're interested in, which might be H3K4 trimethylated. So it recognises that, and then essentially the antibody binds, and you find a way, it can be magnetic or other ways, of pulling the chromatin down after you've fragmented it using different methods. You can do it by sonication or enzymatically or whatever. And then what you do, so you've got this stuff pulled down, and then you elute off the DNA chromatin and you sequence it. And you can say, well, that looks like that DNA was bound to, or chromatin associated with that DNA was bound by H3K4 trimethylated. But now there are better methods, and I want to talk about them if I can. Yes, that's a good point. These methods are generally applicable, not just applicable to embryos, but they're very useful here because some are becoming very sensitive. One of them is cut and run, and you'll still see cut and run mentioned. And in this, the antibody, for example, against our modification or transcription factors that you might think are bound there, it targets, is bound to micrococcal nuclease is actually fused to the antibody, is fused to micrococcal nuclease. So when the antibody binds, it cuts the DNA nearby. Okay? So I got that wrong. The antibody binds, and then you're using something that's protein A that's fused. Sorry about that. The protein A, which is fused to the micrococcal nuclease. And protein A binds to the fragment crystallizable portion of the antibody, which is IgG. Okay, so your antibody goes on, and then you set onto where the transcription factor is, or your histone, your modified histone. Antibody goes on, and then you've got this protein A which binds, this is from Staphylococcus aureus, actually protein A, it binds to FC portion, and then that protein A is linked to micrococcal nuclease, and the micrococcal nuclease just leans over and cuts the DNA out of the side. Okay. Then what you do the same kind of thing, you can purify and sequence it, so work out what's there. So that's an okay method, and it can give you a much. The point is here that it increases the signal-noise ratio compared to chipset that we started, the forerunner chipset that we started talking about. So that's good, obviously. These methods also that you might want to be aware of, assay for transposase accessible chromatin using sequencing, attack set, that's how you'll read about it, attack set. And this reveals genome-wide chromatin accessibility. So it largely replaces DNA hypersensitivity if you read about that. Okay, now this method is increasingly replaced by something called cut and tag as opposed to cut and run. So cut and run, we have the micrococcal nuclease. Here, and it's called, this stands for cleavage under targets and fragmentation, because first of all, the protein A, it's the same principle, but the protein A is now fused to a hyperactive TN5 transposase. So, okay. And what that does is simultaneously, this is a mutated transposase from TN5, Kanamycin transposon from class 2 transposon from bacteria. So what that does is simultaneously cut so the antibody binds, the protein A binds the antibody. Now the TN5 mutant simultaneously cleaves the neighboring DNA and adds adapters, so it's ligation, so that's the tagmentation. Ligating, you flood the system with these adapters. So these adapters can be used for paired-end DNA sequencing, so they increase the sensitivity. And what that does is improve the signal-noise ratio even more, and it makes the method really incredibly sensitive. So now you're starting with this method, cut and tag. You're starting to be able to get down towards single-cell analysis. So that's exactly where we eat, right? Because we're interested in one-cell embryos and early embryos. So that's on the protein, the histone modifications. But there's also DNA methylation, that's another kind of modification, and that occurs via DNT, DNA transferase during gametogenesis. But after fertilization has occurred, many of these methyl groups are removed. In the maternal genome, they're removed passively, but they're removed actively in the paternal genome. So there's active demethylation, and which doesn't occur in zebrafish or xenophus, but it's conserved in mammals. And so the active demethylation isn't really a demethylation per se, it's kind of indirect, and it's indirectly via oxidation. So the methyl moiety, the cytosine, is oxidized, and then the oxidized version, derivative, is then removed. And how is it removed? So that's what I just said, so it's actually oxidized there. So these are what these different adducts look like. And it's removed, so the oxidation occurs by these TET enzymes, 10-11 translocase enzymes, and these are different adducts that are produced. So oxidation, and the one that's present in eggs, there are 3 actually in cells over here, ubiquitous, but the one in eggs is TET3. So that's the one that's only present there. If you make TET3 functional knockouts of mice, so they don't have this oxidation process, then you get a marked reduction in the fertility rate by 80%. And then these oxidation products, when they occur in wild type, are removed by thymidine D-glycosylase or the base excision repair pathways. So they can be removed by standard ubiquitous pathways. And also 5-hydroxymethylcytosine, which is the principal target disoxidative cytosine removal, is an epigenetic marker in its own right, which is still being studied. And this requires other stuff as well. And imprinted genes, I should say, are also marked by DNA methylation in many cases, and they're protected from this demethylation. Now, when I say in many cases, it's not true that imprinted genes are always marked in this way. So the first parents have imprinted genes where they're expressed in a parent origin specific or dependent way. Okay. So usually, so the first parent of origin specified epigenetic imprint marks were DNA methylation. And the reason for that is they were discovered in the, I don't know, well, I suppose they were starting to be mapped to the chemistry in the late 1980s, early 1990s. But it was relatively easy to do because DNA methylation is relatively stable. So that could be done, and it was done. But actually, it turns out that most imprints do not correspond to DNA methylation. This is a paper we published, which I'll just talk about very briefly. So, so many alleles are expressed. So this is a map or a kind of a metric of whether a gene is imprinted or not. But many of these imprinted genes, neither allele neither of the paternal or maternal allele is DNA methylated. Instead, it seems to be marked by H3 K27 trimethylation, one of these marks that I'm kind of emphasizing. So 90% of them are on the maternal allele and 10% on the paternal allele. I'm sorry I'm going quickly here because I've got quite a lot to get through. This is it's cited at the end, but this is the paper in Nature Communications where we describe this work. So this is me, and this is with people in the end. Smart people in the end. Okay. So that's all I want to say about imprinting, and I'm just going to go on quickly now and talk about the really RNA, what's going on as far as transcription and the RNA in the cell. So the oocyte contributes maternally inherited RNA, maternal RNA. As I said, it also contributes many other things. RNA is one of them. And the sperm, although it seems to contain RNA, there's no evidence that it actually introduces any into the egg at fertilization. And that's something which I have a battle with the rest of the scientific community about because they simply haven't shown it, but they write some fancy papers about it. It's ridiculous. So translation of RNA is affected by polyadenylation, of course, generally, but particularly in the one-cell embryo. If the 3' ends are poly-A is polyadenylation, these poly-A tracks that occur in mammalian RNAs, messenger RNAs. If the 3' end is polyadenylation is quite short, 40 to 80 nucleotides, there's very limited translation. But the embryo controls the amount of translation for any given transcript by lengthening the poly-A tail. So the longer it is, the more likely it is to be translated for the mechanisms that I'm not going to go into here. So once it gets to about 250 nucleotides, translation occurs, and this is something called recruitment in the egg, and it's controlled by cytoplasmic polyadenylation elements. So this is obviously something that's not necessarily occurring in the nucleus. And also maternal. This is something this is something that occurs in. Sorry, things crashed. I know it's too good to be true. We're circling for a landing when we were into here, so. I'm going to skip that. I'm going to launch in here. If we can. And if it crashes again, I'll just on the screen mode. All of these changes, I would be talking about imprinting, but in general terms, these changes are involved, many of them, in controlling transcription. It's all about regulating gene expression, and that's particularly important if you're launching a new organism, basically, which you are in a one-cell embryo. And as I said at the beginning, there's no transcription from the gametes. Instead, what's required is activation of expression in the embryo, and that's called embryonic genome activation, or EGA. And what we've done is to use micromanipulation to make precisely synchronous embryos, mouse embryos, and then do deep RNA-seq. So with Brian, who's coming to talk tomorrow. Right. So this is a time course, and we've been able to show that embryonic genome activation initiates, in the mouse at least, within 4 hours of fertilization. So that's a fairly new thing, and it involves all the things that it says there. Canonical splicing, it's a program of gene expression. And actually it turns out that the transcription program is regulated by oncoproteins. So the physiological role of things like SNP, which of course plays a role in cancer, might be to switch on the genome in early embryos. So there are subsequent waves of gene expression. So what the first one we've termed IEGA, immediate embryonic genome activation, which starts within 4 hours in the mouse. And there's minor EGA and major EGA, and we think that minor EGA is a continuation of IEGA, and we've recently reviewed this. So you can read about it in the review we published a couple of months ago. It's cited at the end of it. So here we are. This is the onset of EGA in the mouse, and in human we suggest, and then major EGA, which starts at the 4 to 8 cell stage in human, is here. So the timing is a bit different, and actually we've again published a paper on that, which I'll come to. So these things I'm not going to go into because I haven't got time. There, if you want to ask questions about them, they're there. And this is the paper on human embryonic genome activation, which we published quite recently in Cell Science. The final thing that I want to talk about is the last example of the establishment of totipotency, which has a major class of its own, which is nuclear transfer cloning. So, and to do that, I just want to do a brief kind of contrast and compare. And this is something you might think about, actually, because by doing this contrast and compare, you can think about the things that are going on really in fertilization. What is it about fertilization that engenders this establishment of totipotency? And in the mouse, it's with about 100% efficiency. It's extraordinarily efficient. They shoot, they score. Right? So that's why I put 100% here, I'm not exaggerating. So with that, sperm, oocyte bind, all the things that we've been talking about, second, reduction of meiosis, all these things, they're there. Now, in somatic cell, this somatic cell nuclear transfer, what you do is you first of all remove the maternal chromosomes, so it's called enucleation, from the metaphase 2 oocyte. We're starting with a metaphase 2 oocyte. That's an enucleated metaphase 2 oocyte. And then you inject the somatic cell nucleus into that enucleated cell. So this enucleated cell has got mitochondria, it's got mitochondrial genomes, but it has no nuclear genome in the enucleated oocyte. You replace, therefore, what you're doing is, in effect, you're replacing the maternal genome with the genome of a somatic cell. Okay? And that somatic cell comes from the individual that you want to clone. You want to make a kind of a genomic copy of, a genetic copy of. So you introduce that, the nucleus, and then think about it, all the reprogramming, all of these things that have got to go on. You haven't got to get the decondensation, recondensation, because there are no proteins there. And then you activate, there's no activating signal. We talked about that last Thursday. All of the cell cycle stuff, is it the same or is it different? Well, some of it's different. There's no phospholipase C zeta, because you induce the resumption of meiosis now in this reconstituted metaphase 2 arrested cell by introducing the cell to, in the case of mouse, strontium chloride. It doesn't work for all species. In humans, you might use ionomycin or calcium ionophore or an electric pulse. But somehow you have to kickstart the developmental process, the resumption of meiosis in this newly reconstituted cell. So there are all these differences. It gives rise to one PN, usually, one what we call a pseudopronucleus in the case of mouse, and then it develops an efficiency which is pretty woeful of 2%. There's a very recent paper which has been published in one of the Nature journals, I think, where they claim they can get this up to 30% using tetraploid. For those of you who are aficionados or interested in this, they're using different tricks, including tetraploid complementation, and it's a complete con. but we can, if we're interested, we can talk about that another time. So that's it. I think that's all I've got to say. And you'll read to here. Yeah. So this is a summary of nuclear transfer cloning, and it talks about some of the facets of nuclear transfer cloning. But I just want to actually talk about this in the final minute, talk about this model that seeks to put all of this stuff together in normative fertilization. Sperm comes in, fuses, introduces phospholipase zeta, which indirectly causes a change in calcium. That calcium increase in available calcium activates calmodulin-dependent kinases, CAM kinase 2 gamma, and that promotes meiotic exit, but I think it might also activate transcription factors which become phosphorylated and active. Once they do that, they can participate in remodeling because there are going to be chaperones involved. Of course, in all of this in the paternal genome, there is removal of proteins from the sperm, but basically you have this going on. There's expression of the genes, there's switching on of transcription, EGA, and the transcripts are canonically spliced, and then they start to give rise to mature RNA that gives rise to DNA regulation, cytokinesis, and all the things that then are downstream in combination with the maternal factors that have been inherited from the egg. Like I said, those maternal factors are necessary but not sufficient for the genesis of the embryo, and so on. So that's all I have to say. There's lots of references if you're interested. I'm always available at Thanks ever so much. Sorry about last time, and all the best.